morning. I'm John Engler. I'm with the USDA Natural Resources Conservation Service. I'd like to introduce our next speaker. Dr. Diana Cox Foster is a research leader and entomologist at the USDA Agricultural Research Service, Pollinating Insects Research Unit in Logan, Utah. Her work focuses on biology, management, and systematics of all bee species, with research focused on bees and their interaction with pathogens and stresses like parasites and pesticide exposures. And this morning she's going to talk to us about pesticides and pollinators.
So I'd like to thank Lori and Kelly for the invitation to talk here, talk about my own research. So for 4 years now, actually came about 4 years ago, I transitioned right before NAPPC to go from Penn State University over to the Logan Bee Lab, where I have a great honor of having a large, we're down 2 here, one of them being here in the audience. It's Jamie Strange, who transitioned over department head at Ohio State, but a great crew of scientists there. So what I want to talk about is sort of carrying forward, take you to another class of chemicals that you may not have heard about as much. So our lab, we are the home of the U.S. National Pollinating Insect Collection, and it's one of the largest collections of bees throughout the world. And as you know, bees are incredibly diverse. We have 20,000 or so species globally. Yeah. And in North America, we have over 4,000 species. And it turns out many of my scientists who are systematists tell me that we're going to be coming up to that. They tell me they have all these drawers full of undescribed species that they're continuously working on. One of the things that's ill appreciated is that honeybees have been distributed globally by us as humans. They weren't native to the United States or North America to begin with. We have bumblebees, but the vast majority of these 4,000 species are solitary bees. We now know that, indeed, that there are several bee species that have been declared to be endangered or critically of concern. We still don't know what the vast majority of those solitary bees are doing, but we do know that there is a decline. So with honeybees, we know that we still have extraordinary losses of bee colonies, up near 37 to 40% loss of colonies annually by beekeepers. For the bumblebees, we know that we have these declines going on as well. So what are the causes there for the pollinator health? And as being one of the researchers that's been tied up into this work, I can tell you that there's been a lot of controversy and dispute about it, but I think you can summarize it down to these 5 main factors: pesticides, pathogens, poor nutrition, i.e. the lack of floral resources, parasites, and now we understand that climate itself sort of goes across the whole board here. The critical thing is that we now know that these all interact together, that there's not one silver bullet out there that we can pull apart and dissect. When I was a faculty member at Penn State, I was more on the pathogen side, but I had a great set of colleagues, Chris Mullen, Marianne Fraser, Jim Fraser, who have now all retired, and started the focus on the pesticide side. We now know that pesticides move globally through water and air, widely distributed, and that, yes, indeed, we can go back and find chemistries that should have been long gone when I was a kid, like organochlorines, that those are there. But one of the things that Chris Mullen also pointed out is that in terms of chemicals, there are other things that are being used, and these are adjuvants. And so what are adjuvants? There are things that can be included in formulation. In the United States, it's a trade secret still what those chemicals are. But there are other things that growers and farmers, landscapers, i.e. people doing lawn care, add into their tank mix to help make things work better. One of the group of these is the organosilicone spray adjuvants. And these are polymers that have silicone included in them. They have different modifications, chemicals, so they come in different flavors. They're widely used not only in agriculture and landscaping, but they're also widely used in industry, medicine, like lubricants for syringes, Cosmetics, many of your hair care products that you use today had these in them. And then I lastly learned they're also used in the manufacture of artificial snow. So many of those ski areas are using these to produce their artificial snow. So even in more pristine areas, these are being out there. In agriculture, they're included to supposedly increase spray coverage and uptake of spray solution in plants. And there's evidence for that. They're included in several patents to deliver RNAi into plants to get it in there. And I was surprised to even find that some of them have received certificates for restricted use by the Organic Materials Review Institute, i.e. this is where growers can go to try to find things that to use for organic farming. And unless you look closely at the certificate, you may not see that restricted use part in there. So just give you an idea what they do. This is showing an herbicide applied on the left-hand side here with a different type of adjuvant, and on the right with the organosilicone. You can see on the right that it's spread out, covering much more of the surface area there. And these are also super penetrants, so things get into the plant tissue very quickly. Chris Mullen did a lot of work on this, and he was very patient to go through the California database on pesticide usage. And he, through his work of students, showed that these could impact learning in honeybees and were highly toxic. So at the top here in this graph, there's a purple line at the very top. This is control, where the bees are being fed sugar water. The other lines here where they're being fed the organosilicone at 100 parts per million, and the three different types or forms here. You can see that the one form in red here is highly toxic, killing the bees in 2 days. You may say, if you've looked at pesticide molarities and how they're being used part per million, that this is extraordinarily high until you realize in the tank mixes, these are being used anywhere from 300 parts per million to 5,000 parts per million. This is well beyond what the use is in the active ingredients. And down below are the data from the California database. These chemicals were developed in the 1990s, and what you can see is it's an exponential increase. The bottom here, the year out is 2014, and one could go in there and look at this. Over here on the left, the graph here, this is 1,600 or 160,000 pounds is the top line there. This is well swamps anything else. One of the things I learned as a research leader talking to alfalfa seed growers is that they had been routinely incorporating these in each and every spray that they put on their crop. And alfalfa seed production is an intensively sprayed crop 10 times a year. So it far swamps out any active ingredient and goes across all cropping systems, fruit and nut production, vegetables, over into landscape lawn care, take it up to, I think it's 0.4% is where they say that you can incorporate it in. It becomes phototoxic after that, and ornamentals. Herbicide use, this is something that's routinely added into herbicide use. So in terms of exposure to bees, this is something that would be found in many environments in many areas. So I found with Chris Mullen retiring, he was the only person doing active research on this and having ability to do assays. I was lucky to find an environmental chemist at Utah State University, William Doucette, who's in the Department of Chemical Engineering, who's developed now the assays and improved on them that Chris had. So through funding from the American honey producers, we joined up with commercial beekeepers, and got samples from almonds that came in 2017 from that pollination season. These OSS were showing up at high levels, 45%, 23% in collections of pollen taken there. But the other critical thing is that many of those pollens had a gemisch of pesticides in there, fungicides and insecticides, including ones that were not probably coming in from other areas like mosquito control. So these bees are being faced with not only one chemical, but lots of chemicals. But these organosilicones are there in high amounts. So my own interest comes from the pathogen side, and just to give you a brief introduction there, bees are just like us. They are faced with a wide variety of different pathogens, including viruses. We have now over 40 different kinds of viruses described, and the ones that I focus in on are the RNA viruses, and these have been associated with colony losses. We now know that these are also infecting pollinators more broadly, and that they can be shared back and forth with each other, many of these. So with one of Chris's students now, Dr. Julia Fine, who's now in ARS as well as a scientist, back then we did a study in which she was able to grow up honeybee larvae, and we added into the in vitro assays the OSS by itself, a little bit of viruses. Honeybees, when they're infected, the nurse bees, they actually secrete viruses out, just like you and I do when we have cold and sneezing and whatnot. And then we combine the two together. So we went from 25% mortality to over 75% mortality when the two were combined together. They synergized to get this increased mortality. And many of the bees died as they're metamorphosing. And this matched the symptoms that beekeepers were describing, seeing in their colonies, bringing them out of almonds. To our surprise, there was a virus that was associated with this increased mortality, and it's a black and queen cell virus, and it's one that you may not have heard much about if you talk with beekeepers and whatnot. The distinction of this, it went from being fairly rare, back when I was doing studies at Penn State in the early 2000s, to now being found in over 90% of honeybee colonies, and also being commonly found in bumblebees and other pollinators. It's the one that increased and was associated with this loss. We also saw that there was a gene involved in antiviral immunity that did not turn on. It was suppressed by the OSS. So we've carried that forward into doing a comparative study. And one of the things now is to think about how honeybees compare to the other bees, bumblebees and solitary bees. I'm going to put forward that honeybees actually are much more resilient. If we're having issues with honeybees, then we need to be even more concerned perhaps with the others. So one of the things that honeybees do is that all the food handling, the processing is done by nurse bees. They're the ones who take the pollen and nectar and break it down and create proteinaceous secretions to feed everyone else. Doing this, they detoxify those chemicals coming in, so the others are getting relatively clean food products. They're not having to deal with that pesticides or these contaminants directly. You also, if a healthy colony, you have a large number of bees in there. You can afford to lose a few thousand a day because they're being constantly regenerated. The one key individual in that colony that's needed is the queen bee. She's the mother of all. So her health is very important. We now know that beekeepers say that queen bee health is a major issue. It used to be, I think, when I was a kid and even as a new faculty member, queen bees would live up to 8 years. Now they're lucky if they live 1 or 2 months, which is dramatic. So we know that from other research that chronic impacts may remove the resilience. One of the issues in going from doing bioassays on single bees to colonies is that this resilience prevents you from seeing a lot of things in the short term. So one of the things that we did in Logan for my research is we took and got rid of that resilience. We took colonies down to micro sizes here. And there's a guy down in Salt Lake who makes these cute little colonies that are good for your backyard beekeeper, but they're also good for experiments where they're greatly reduced. We got sister queens, we marked them, we clipped their wings, which were important, and then we fed these colonies OSS or pesticide in there. This was funded by the Almond Board, so we did a fungicide and insecticide as well, and then a combination. Randomized them, and we looked at brood over time and the bee counts and monitoring the queen. We opened up these colonies after a week, and it turns out some queens were much more attractive, so workers moved over and gave us a greater reassortment. We had really dinky, tiny colonies and some bigger size colonies as well, intermediates. So the different size or the different shapes of the symbols here indicate the size here. So what we found was the small colonies had really hard time, but some of the control colonies survived. So each of these lines here, individual colonies, and the dots going up indicate what happened over time. One of the things that we saw with the pesticide, both the fungicide and insecticide treatment and the OSS, is that the queen health was impacted. So we saw that either the queen disappeared, died out completely, or this little queen with her wings clipped got up and moved out of the colony with her bees, finding them out in the ground. They're in my backyard, so it was easy to find this little swarm crawling across the area. We modeled that, and it turns out that, yes, indeed, treatment was important, but size was also important. And as you'll hear later from Tim Mays, is that size of colonies is now one of the things that we see changing, that even all these bees going to almond board or to almonds for pollination, this colony sizes are getting reduced because it's just really hard to get those really healthy colonies up and going. So we are having this impact. We also saw in the viruses that the viruses were increasing by between the period of time here, that the OSS having this impact on the colony and on the workers themselves. And I'm not going to tell you about it, but we've gone on to do transcriptomics and looking at what's changing in these bees and that. If you have questions about that, I'd be glad to tell you about it. The critical thing is that this is not just a honeybee issue. There's impacts on other bees. For bumblebees, we've found that these are highly toxic to bumblebees as well. Looking at Bombus impatiens as our model bee here. So feeding them 100 parts per million of the OSS in sugar water kills them very quickly as well. We've gone, though, on to solitary bees. And for this, we've used, as our model here, the Alfalfa Leaf Cutting Bee, Megachilodendrota, which is a bee that's used for commercial pollination in alfalfa, and now has also been shown to be able to be used in canola, blueberries, and other crops. And the key thing here is that this is a solitary bee. You have to think of solitary bees as the single mom. She's all on her own. She's out there collecting the pollen and the nectar reserves. And these bees also create their cavity nesters, so they take leaf pieces. So they're one of the ones that go out there and do like a leaf punch or a hole punch and punch holes out of leaves. She creates this little cavity of leaf pieces, puts the pollen and nectar in there and lays an egg. You now have added risk of exposure to the pesticides. There was a meeting in 2017 convened by industry with many regulators, and NAPPC was helping to lead it. We all agreed that yes, indeed, bumblebees and the solitary bees face this added risk here. So the larvae directly eat that pollen nectar. They don't have any ability to have it detoxified before it gets there. There's potential for that leaf pieces to have been contaminated with the chemicals themselves. So what we did was we set up experiments to dissect these out, to pull them apart and ask what was important there. We did this in cages, and we did it with two plants. One was Phacelia, which gave them pollen nectar, and they don't make use of the leaf of that plant. And we gave them buckwheat, which they like the plant. You can see the two plants down on the bottom right here. And we just asked about OSS here. So we either sprayed the plants with water or sprayed one or the other with the OSS or sprayed both of them together. And we did this at the concentrations that an alfalfa seed grower would be doing and on the same spray schedule. So one of the things that we found was that, yes, indeed, that this OSS had an impact. We're still looking at the viruses and the parasites there. But what we found is it affected the behavior of the bees. Those females that got exposed to the OSS and the pollen nectar formed 60 to 50% fewer nest cells. That was a great reduction in her reproduction. I think I said that right. And we had these bees marked, so we knew who was doing what and what their responses were. So what I told the beekeeper or the alfalfa seed growers, to put it in a different way for them, to shift it from bees back to their crops, they're hiring these bees. They're actively making use of them, buying these bees, growing them up, and that's to do a job for them, and they want the pollination. This 50 to 60% reduction in pollination translates into a lot less seed potentially for them. And I said, if you had 2 workers and one was doing a lot less work, 50% less work than the other person, would you be happy? So it's not really clear that the OSS is benefiting them in terms of their pest control or what they're using it for. So to take this back to another level, as a graduate student at University of Illinois, I didn't think at the time, because we were required to do all these core courses, that we were required to take courses. And one of the people I got to take integrated pest management from was Bob Metcalf, who's a National Academy of Science member. And his journey as a scientist is that he moved from being pro-insecticide, working in California, UC Riverside, out in the fields, actually having to get atropine shots because of the insecticide exposure, to becoming a proponent for integrated pest management. And just to show you this here, this is written in the 80s, 87, saying that we became too focused in on pesticides being our first line of defense against pests. He said that we needed to go back to integrated pest management, where pesticides are still part of the arsenal to try to combat pests, but we need to bring in other ways to control the pests there. I would say that now, 30 years later, we're still at the same situation here. And so other colleagues at Penn State have put forward that we need to add another P into IPPM, Integrated Pest and Pollinator Management. That we need to think of controlling the pest, but promoting pollinator health at the same time in those crops. And so with that, I'd say that we still have this concern overall for all the bee species, solitary, bumblebees, honeybees, for the impacts of pesticides and these adjuvants. And it's, I think, very clear from our work with bees, and there's many other papers out there, including thesis work done by Mary Purcell and Larry Chandler, who's one of the area directors of ARS, that these OSS are insecticidal. They're actually used also for hair lice and sand flea control, which is a major pest in Africa. So this does have a big impact. So recommendations here is that we need to find other adjuvants if it's needed, and we need to limit the OSS to sprays when the bees are not there or apt to be introduced, and definitely go to integrated pest and pollinator management. So I'd like to thank all the people that helped me to get the work done here, and the Almond Board for part of the funding on the studies. And thank you for inviting me.